The overall goal of this protocol is to use a single combination real-time assay to identify specific small molecule inhibitors of intracellular bacterial growth that are non-toxic to host cells.
This method can help answer key questions in the antimicrobial discovery field. Specifically, what types of compounds can penetrate into intracellular compartments and kill pathogens without harming the host cell? The main advantages of this technique are that bacterial growth and mammalian cell toxicity are detected simultaneously using non-destructive real-time assays.
Generally, individuals new to this method will struggle with the scalability that it affords. Assaying greater than 10,000 compounds in a session requires a whole new set of techniques and equipment.
In addition to Lucius, demonstrating a communal commitment to academic antimicrobial discovery and translational diagnostic microbiology are postdoctoral fellows K.P. Smith, Yoon-Suk Kang, Jennifer Tsang, Thea Brennan-Krohn, and undergraduate Kat Truelson.
To prepare host cells, culture J774A.1 macrophages in suspension in RPMI 1640 with 9% iron supplemented calf serum. Initially passage cells in tissue culture flasks. After cells have become confluent in a 75 square centimeter tissue culture flask and 15 milliliters of medium, split the cells by scraping and diluting them to 65 milliliters with the